In the winter of 1972, tourists were flocking to the beaches of Hawaii, but a few blocks away from the sand, a scientific conference on bacterial plasmids was taking place, and a brief conversation between two researchers there would lead to the first successful construction of artificial recombinant DNA. Stanley Cohen, a researcher at Stanford, had been studying plasmids, tiny, free-floating rings of DNA inside bacteria that replicate independently. He met Herbert, a biochemist from San Francisco, who had recently isolated specific enzymes capable of cutting DNA strands at precise, predictable locations. Over deli sandwiches, they realized their individual projects were two halves of a single process. Boyer's enzymes could open a DNA sequence, and Cohen's methods could insert a foreign gene, like one for antibiotic resistance, into a plasmid, which could then be reinserted into a bacteria to replicate. By successfully isolating and multiplying foreign DNA in a host cell, Cohen and Boyer moved biology past observation. They provided the mechanical steps for cloning genetically engineered molecules, which served as the technical foundation for the global biotechnology industry. Humans have actually relied on biological systems for thousands of years. Making curd, baking bread, or brewing wine are all microbe-mediated processes used to create products for human benefit. But today, the European Federation of Biotechnology uses a much more specific definition. They describe it as the integration of natural science and organisms, cells or molecular analogs, for products and services. This era relies on two core principles. The first is genetic engineering, the use of techniques to alter the actual chemistry of genetic material, like DNA and RNA, to change the physical traits of a host organism. The second is bioprocess engineering. Once you have an engineered cell, you must maintain a sterile, contamination-free environment on an industrial scale. This allows the desired microbe to grow in the massive quantities needed to manufacture vaccines, enzymes, or antibiotics. Traditional hybridization in plant and animal breeding has a major limitation. When you crossbreed to get a desirable trait, you often multiply undesirable genes alongside it. Genetic engineering overcomes this. It allows scientists to isolate and introduce only the specific, desirable genes into a target organism, making biological development a predictable technology. To successfully modify an organism, a researcher must execute three mandatory steps: identify the DNA with the desirable genes, introduce that DNA into a host, and ensure the host maintains and transfers that DNA to its progeny. Because a piece of alien DNA cannot usually multiply on its own, it must be linked to a delivery vehicle. In biotechnology, we use plasmids, circular extrachromosomal DNA, to act as these vectors. Isolating target genes from the source DNA requires restriction enzymes. These act as the biological tools needed to snip DNA strands at highly specific locations. These enzymes are categorized by how they cut. Exonucleases remove nucleotides from the ends of the DNA strand, while endonucleases make precise cuts at specific points within the DNA molecule itself. Restriction endonucleases scan the length of a DNA molecule until they find a specific recognition sequence. Once found, they bind to the DNA and cut both strands of the double helix at exact points in their sugar-phosphate backbones. Today, researchers use more than 900 different restriction enzymes, isolated from hundreds of bacterial strains. Without these tools, we would have no way to isolate the specific biological instructions needed for genetic recombination. The sequences these enzymes recognize are palindromic. Just like the word Malayalam reads the same forward and backward, a palindromic nucleotide sequence reads the same on both DNA strands, when the orientation, the 5' to 3' direction, is kept the same for both. EcoRI identifies the palindrome GAA-TTC, cutting both the vector and foreign DNA slightly away from the center. This leaves overhanging single-stranded sticky ends primed to form hydrogen bonds. When they meet, DNA ligase joins the fragments into a recombinant DNA molecule. By using the same restriction enzyme on both the source and the vector, we create matching biological docking ports. This allows us to merge DNA from completely different species into a single functional unit. Creating a recombinant molecule requires preparing two different sources simultaneously. On one side, we isolate the foreign DNA containing our desired gene. On the other, we isolate the circular plasmid that will serve as our vector. Both are treated with the exact same restriction enzyme. This ensures that the opening in the plasmid vector and the ends of the foreign gene share the same staggered sequence, allowing them to fit together. DNA ligase is then used to fuse the foreign DNA into the plasmid. This creates the recombinant DNA molecule, a new genetic combination that does not exist in nature, but is now ready for use. During the transformation phase, this recombinant plasmid is inserted into a host cell, such as E. coli. Because E. coli is closely related to the original source of many plasmids, it can readily accept and maintain the new genetic material. The recombinant DNA includes a specific sequence called the origin of replication. This forces the host cell to duplicate the foreign gene every time it divides, turning the bacteria into an automated factory cloning the desired DNA. As the bacteria multiply and follow the instructions in the new DNA, they begin producing specific proteins. This is how we produce vital medical products on a global scale, such as human insulin, which is harvested directly from these engineered biological factories. The meeting in Hawaii provided more than just a new experiment. It provided a workflow for turning microscopic cells into programmable manufacturers. By mastering the ability to cut, paste, and replicate genes, we have redefined our ability to produce the medicines and foods that sustain human life.